So I'm going to assume everyone can see them since you guys can't talk back to me. But this webinar is on clinical trials one-on-one. I'm Sydney, for those that don't know me. I have a PhD in pharmaceutical sciences. I'm also the director of TGCT support programs, and I work with the Life Raft group to try and make resources for our small little disease. And this webinar is supported by an educational grant from DSI and Decipher. So I like to keep it in a very classroom looking appearance, because that's really what it is. Most people have never heard of clinical trials or have any experience with them. So this is, for most people, the first time. And unfortunately, the first time that they've had to go through an illness at the same time. And so to start out, we have to get to the same basic definitions. My pointer's going a little nuts, but what is clinical research? Clinical research is defined as many different types of research. It can include epidemiology studies, which improve the researcher's understandings of a disease and its course, behavioral studies, which allow researchers to learn how behavior impacts health. This could be whether smoking impacts your life, how much exercise should you have on a daily basis, those kind of things, but they're not interventional, which means that no one's assigned anything. There's also health services research, which is how we understand access and healthcare costs and how that impacts individual patients. But overall, why we're here is more about clinical trials, which is a form of clinical research where we test a new way to prevent, diagnose, or treat an illness. And this may be by using a new drug or a combination of drugs. This may be a new way of doing surgery or a new device for surgery, injectables for insulin, any form of medical device, or new ways of treating for an existing disease already. And so clinical trials are conducted according to a plan. It's very outlined, it's very systematic. It's not fly by the seat of your pants by any means. And so this is called a protocol. And these protocols specify for each study what type of patients should or is perfect for the trial, the schedule of the tests or procedures that are conducted, the drugs involved, as well as the dosage and the amount of drug, meaning the duration of treatment, the length of the study overall. So you may only receive treatment for a short time, but follow-up may be for a really long time, or the screening process to find out if you're someone who should enter the trial may be excessively long. So the duration of the study is different from the duration of your treatment. It'll also include things like what side effects would warrant you to stop the drug, as well as maybe some risks and side effects that you would expect, as well as overall what researchers hope to learn from the study. And so those protocols can be hundreds and hundreds of pages. Mostly what patients see are this informed consent form. And informed consent is a process in which patients are given adequate information, including information on the possible risks and benefits about the clinical trial, to inform their decision-making and their active participation in it. And this infers through this informed consent process that patients have sufficient information on the benefit, predicted information on the risks, the requirements to fulfill the study, how many visits, how often you have to be there, how many times can you miss a dose, things like that, the duration of the study, the purpose of the study, procedures within the study, outcomes of the study or potential outcomes, with sufficient time to discuss with your friends, family, and other providers. At that point, a patient can verbally or in writing make an informed decision on their participation. For all of us, we would sign an informed consent form. And most importantly, that participation is voluntary. And at any time during the process of the trial, you can withdraw your participation without any consequences or penalty. And that's across the board with any form of clinical trial. So we break clinical trials down into specific phases. You have phase 1, phase 2, phase 3. After phase 3, a drug will or won't be approved. And then phase 4, which is post-market research. And this phase 1 through phase 3 process takes around 7 to 10 years. So a drug from the inception of taking all of those particles, making them, compacting them into a drug, testing them in animals can take anywhere from 12 to 15 years. And it's said that only 1 in 1,000 compounds will even make it to a human. So if you can imagine, that's a lot of trial and error at every aspect of the trial process. And so we'll go through each of these phases. So starting with phase one, the main purpose of phase one is whether the investigational treatment is considered safe. This is typically done in healthy volunteers because it lessens the amount of variability. And it's done in a very small amount of people, usually somewhere around 10 to even 50 people. And that is how we identify what the side effects are, whether the side effects are reversible, what dose is of interest and what dose will be given, what is the safe dose, is it safe to take with additional drugs? So whether you can even take it with foods like coffee or grapefruit, this is all investigated around this stage. And these trials can take anywhere from 1 to 6 months, with most of them taking around 1 to 14 days of treatment. And so we'll go through some of the features of phase one studies because they're really how we get the dose, or at least the starting principles of what's safe about the drug. And so phase one studies can have many designs, but the most common designs are what we call a SAD or a MAD design. And so a SAD design is to determine the safety of the drug, but it starts out where patients take a single dose at one time. So single ascending dose, SAD. So we'll take a single group of patients. We have group one or a group of healthy adults, and they would receive a single dose, and we can just make up a dose, so 10 milligrams here. And after those patients take, or healthy volunteers take the drug, they're monitored for a short period of time. Doctors and researchers will start to get information on their safety profile, whether they get headaches, maybe some of them have nausea. They write all of that information down. After an elapsed period of time that the dose at this 10 milligrams is deemed safe, another group of people, patients, subjects, whatever word you want to use for this group of patients or healthy volunteers, will receive a slightly higher dose. So they'll receive then a 1 dose of the 15 milligrams in this case. And during that time, the researchers will itemize and figure out what type of side effects that these patients or healthy volunteers have. After that time has lapsed and all of that information is obtained, 7 days is my arbitrary number I give, they'll allow another group of people to receive the drug at a higher dose. And so that, in this case, is the 30 milligrams, and they'll continue to get safety data. And that's how we get safety data from a range of doses. And so this allows us to determine what side effects each dose has, but it also allows us to know what dose would we move forward with. Because if these healthy volunteers or patients have complete terrible stomach GI issues that all of them withdraw, maybe that's not the dose that will be used. The other design type is a multiple ascending dose study, where patients will take the same dose but multiple times. So say in that SAD study, the 10 milligrams wasn't what we wanted, so we went with the 15 and the 30 milligrams, and we really want to identify which of these will go forward. We would take a separate group of people. In this case, you have group 2 and group 3, and you would give them the 15 milligrams or the 30 milligrams every day in this case, or whatever regimen you're trying to test, for 14 days. And so that allows us to know what side effects are each dose, but whether side effects happen cumulatively. So meaning that you wouldn't have side effects maybe the first time you take it, but you may have side effects 3 days after taking it. And this also allows us to pinpoint what dose will move forward for the phase 2 studies. And so within the phase 2 studies, what you're looking at is whether the investigational treatment is safe, but it has some signals that it may work in the intended population. This is the first time in which the drug will be given to patients. And patients, because each of us are very different, our disease may affect how we respond to the drug as well as our general health. This data is a little bit maybe harder to interpret than healthy adults. And so what we get from this study is how should this drug be taken and how often? What is the dose to be selected for the major pivotal study? And how long are these are there long-term side effects associated with the drug, longer than 14 days? And then is there signs that this drug may work? And these trials are typically anywhere from 3 to 12 months, and they have more patients than before. So for TGCT, because we're a really, really small population, you may have 10 to 20 patients in this phase 2 study compared to the phase 3 study where you may have 90 to 120. So you can see how we grow over time with each phase. So if we go to phase 3, which is known as the pivotal study, this is the study in which you're trying to get all of the information that does this drug work, how well does it work, and who does it work best in. So this is focused on the effectiveness while making sure you capture any safety data. So how effective is this treatment? How does it compare to other treatments that are out there? What is the optimal use and in what population? Maybe there's a specific group of patients that it works best in. And is there a benefit or a risk to a specific population? And so often this is where you start to develop the idea of who will the drug be marketed to. And this can be anywhere from 12 months or much longer of duration. And this often has many patients in it. For TGCT, it's somewhere between 90 and 120 patients. But for more common diseases like cardiovascular related diseases or common cancers like breast cancer, you may have thousands of patients enrolled in these. And for people who follow maybe the vaccine trials for the mRNA-based vaccines had 30 to 50,000 patients in these trials. So you can see that it grows depending on your patient population. And while those phase 1 through phase 3 trials seemed very distinct, they seemed like maybe there was no overlap. Often you have to decide in a rare disease population how best to use patients. You don't want to use all your patients in the phase 2 trial and not have enough for the phase 3 trial. So in that way, you can combine parts. You can have a phase 1 and a phase 2 trial combined, or a phase 2 and a phase 3 combined. And that allows you to obtain as much data as possible from the population. But while those studies are being conducted, there's many more studies happening. There's not just 3 studies in patients that happen over the course of those 7 to 10 years. There's a lot more information. And so often we have to answer questions like, can the treatment be taken with food? And we would do that with a dedicated food study where patients or healthy volunteers are given food at different intervals, and they see how that changes the drug concentration through taking blood collection at different time points. Can an individual with liver or kidney impairment take this disease or take this treatment? So often TGCT specifically, it doesn't impact our life expectancy. We're going to live the same amount as maybe the general public will. And that means that we may have other diseases like liver and kidney impairment. And so can we take these drugs still if we're someone with other diseases? And so through studies of enrolling people with liver and kidney impairment, we can find out based off of how a drug is eliminated and whether someone needs a dose adjustment in their dose based off of if there's someone who has impairment that's mild impairment versus very severe impairment versus on dialysis, for example. There's also studies that are conducted to find out what drugs interact with the treatment. And this is often done with preclinical studies with other drugs where you use cells or animal models to determine whether drugs will interact. Is a treatment toxic to a fetus? This is also done using preclinical animal studies to determine what type of toxicity it does have and if it affects pregnancy. And lastly, but not most least, is that should doses in children be different than doses in adults? Children are not just shrunken down adults. They often have their own considerations. Their enzymes in their body mature at different rates than ours as fully matured adults. And so often modeling and simulation is used to determine what dose should be used in children. And so some features of phase 3 studies that you're probably pretty familiar with at this point are placebo, randomization, blinding, crossover. And some of these different features of a trial may be used in phase 1 or phase 2 to increase the scientific rigor and get more answers earlier on. And we'll talk about a little bit on why, because someone asked a great question on why. But we'll start with the language of what does these all mean. So starting with a placebo, A placebo is an inactive substance, often a sugar pill, that looks identical to the investigational treatment. If it's an IV or an injectable, often it's just a saline injection. But the main point is that it looks identical, but it has no active ingredient in it. For TGCT, there is no standard of care, which means when we're diagnosed, there's no best way to treat us. We're very patient-centric. It depends the person. And I think we're all tired of hearing it depends, but it's true. And so for us, placebos are commonly used in these trials, but for diseases where a standard of care exists, that standard of care can be used as the comparison group. For us, because we do have a placebo, or because there is no standard of care, often the placebo is used to compare directly to the investigational drug to see if the investigational drug works. And this is largely because a person's expectation of whether they will get better impacts, honestly, if they get better or not. And those who receive more care and attention in a clinical trial may start to get better inherently because they're receiving more care. And so with that in mind, there has to be a way to control for biases in people feeling better, just by feeling better. When you feel like your needs are met by when your care team truly is caring for the holistic person, which sometimes happens in a clinical trial, that reduces stress. And stress impacts pain. And for a disease that majority of us have some form of pain, it becomes really important to have a comparison group. And so this allows researchers to compare the effects of a drug and the side effects to something. And that the response, so if there is an equal response to a placebo, then that drug would not be considered effective. And so placebos are considered the gold standard in this phase 3 studies and may be used in phase 2 studies to acquire more data to determine if a drug works early on. Randomization. Randomization is the process in which patients are assigned to either a treatment group or a control by complete chance. Usually this is done by a computer that generates a number and that sets you into a specific group, whether to receive the placebo in this case, which is the control group, or to receive the treatment group. This is largely because patients and their diseases are unique. And so if a patient gets to decide what group they're going to be in, there may be patients that are more severe in their presentation that decide the treatment group. And so biases may be presented, or if a provider is the one deciding, providers want patients to get better. And so they may be more likely to put the more severe patients into a treatment group, for example. However, randomization does not infer that you have the equal chance of being in either of these groups. And what you'll find with many of the TGCT studies is that you have a much higher chance of getting the treatment than you do the control. And overall, this randomization is used to reduce bias, particularly selection bias. Like I said, that a patient would self-assign themselves the treatment or choose to go into a treatment group, or the provider chooses a patient to go into a group, because that's very specific. You're picking different characteristics, even if you implicitly, even if you don't know it, to go into a group. And that also reduces accidental bias, which is just an inherent bias we all have, whether discriminatory or not, to put patients even that are similar to us or look similar or have different history in a specific group based off of those characteristics. So blinding is also a type of technique in which patients or their providers, so if a patient is the only one blinded, it's considered single blinded. If a provider is the one also blinded, it's considered double blinded. But ultimately, blinding makes it that the patient and or the provider do not know which drug or treatment is being given. And so randomization reduces the bias that we would select patients that would benefit most from the drug. It does not eliminate this idea of the placebo effect. The effect that if you know you get the drug, you will get better just by the thought of you getting a treatment. And so placebo effect is extremely powerful, and blinding allows no one to know who's on the drug and who's not. So you are not treated differently, or you don't think you're going to get better based solely on that. And so blinding allows us to measure more subjective measurements, like how a patient feels. And so pain being the number one symptom many of us experience, it's important to have blinding So we can accurately measure whether your pain is getting better because you think you're receiving a treatment or because truly the drug has an effect. And the opposite of a blind study is considered an open label study, which you know the label that you're getting. And lastly, we have this crossover effect or crossover design. And this is a type of study that allows patients to cross over into a treatment arm if they've previously been on a placebo. So for example, if we take this 120 TGCT patients, we randomize them in a 2-to-1 ratio, which means out of every 3 patients, 2 of them will receive the drug and 1 of them will receive the placebo. So you have 80 patients receiving the TGCT drug and you have 40 patients receiving the placebo. So again, more patients will receive that treatment up front. But for 6 months, they receive that treatment. They're taking it, in this case, we'll just say daily. And then at the 6-month mark, it's determined whether the drug is working. And we'll talk more about what does it mean when a drug works. But at that 6-month mark, after the researchers collect the data on whether the drug works and whether the placebo has an effect or not, you're unblinded, which means patients are told which drug they were on. Once you're unblinded, most often there's a second period where patients, whether they receive the placebo or the treatment drug, are allowed to receive the treatment. And this is really important for TGCT, where we all sign up to be in a trial where we get better. So it's ethically and morally an obligation to allow patients who just gave their 6 months of effort and time and thinking that they're going to get better to give them the drug. And so this is an extremely important part for patients to know exists. And so some examples. So starting with the pexidartinib trial, which is Turalio for those unfamiliar, it was called Enliven. In their phase 3 design, so their last pivotal study, they took patients that had a confirmed diagnosis and they were symptomatic of TGCT, where surgery would be associated with the potential for their lifestyle, their pain, their situation to get worse just by having surgery. And they had to have measurable disease. And they were assigned 1-to-1, which means 50% of the patients received the TGCT drug and 50% received a placebo. This is less common now that there are drugs available, but the patients were randomly assigned pexidartinib, where they had taken 1,000 milligrams per day, split into twice daily for 2 weeks. And then they received 800 milligrams per day, split twice. So 400 in the morning, 400 in the evening for 22 weeks for a total of around 6 months of treatment. Or they received a matching placebo group. And this was for that 6-month period. At that 6-month mark, which is this slashed line here, everyone received the pexidartinib or Turalio for an indefinite amount of time. So that's why it's called an open label extension. It's an extension to the first part of the trial, and everybody knows what drug that they had prior, and now everyone is allowed to go on pexidartinib. And we'll talk more about kind of these outcomes of how do you know a drug works. For them, they looked at patient-reported outcomes as well as MRI to see if the drug was working and the disease was shrinking. And so clinical trial endpoints is a hot topic. You don't even know what you walked into. But when we say a drug works, that has to mean something. There has to be a value for what that means. And so often endpoints are what we use to determine how a patient will benefit from a drug. This, in the case of TGCT, is often done with MRI measurements, but also questionnaires where patients are asked about their pain, stiffness, and function. Ultimately, the goal is to capture the improvements in how a patient feels, functions, and survives. Shrinking a tumor just to shrink a tumor when no one benefits, there's still the same level of pain, stiffness, and overall impact on quality of life is not an effective treatment, and the patients don't benefit. It is extremely important that there is improvement in how a patient feels and functions. And so many of you know that TGCT is mostly measured with MRIs, usually with or without contrast. But in the trial setting, this RESIST 1.1 criteria is used. And using AI, we can make it look red in these images where you can see some of the tumors, but in real life or in real imaging, there's not a sign that says, look, there's disease. You really have to measure it and be proficient with understanding the radiology. And so when we say complete response, we mean that there is a disappearance of all tumors completely where they're unmeasurable. They're undetectable by eye. A partial response is someone who has 30% or more decrease in the sum of the diameter of the largest tumor. And so that's kind of a weird way to measure things, and we'll talk more about why that way isn't necessarily the best. But this also includes that there's no new tumors and no progression. For stable disease, it would be that there's no new tumors or progression, but there was no shrinkage. And I think stable disease is really underappreciated. And the longer maybe you have TGCT, the more you kind of appreciate just stable, unremarkable. But going back to RECIST 1.1 criteria, which is a way of measuring the disease, it measures the sum of the largest diameter of the tumors, which a diameter is a two-dimensional measurement. And so if we look closer at these, some of them are really easy to measure in that way. So if we look at this knee, I'm going to orient you guys for those not super commonly staring at MRIs. This is someone's knee. So this is the femur, and this is their femoral head, and then this is their tibia. This would be if I was standing sideways and you were looking at me from the side. And what you can see is there's this tumor in the back here. This is the back of the knee, and this is the front of the knee where the kneecap is. And so what you can see is if we take the largest diameter of it, that's pretty easy to measure. And this is probably because the tumor is localized relatively. It's very small, it's compact, it's nodular in appearance. But if we take something like this Honkin disease, it's really hard to determine where it starts and where it stops. Does it stop right here? Does it keep going into this tissue? What about this tumor? You can see how it starts to get difficult to measure. But if we just take this part of the tumor and we take the largest diameter, that doesn't entirely tell the whole picture about what's going on everywhere else. Similarly, if we take this very distinct posterior disease in the back of the kneecap, you can see the same, that you have this large amount of disease spanning from here to here. And it's much easier to see because AI has taken it and made it very red looking. But if it extended a little further, you wouldn't be able to see where it started and where it stopped in the muscle tissue either. So very quickly, this isn't the perfect measurement, but it's the main one we use. So thinking and keeping that in mind, that RECIST criteria maybe isn't the best, and to use some actual MRIs that make it, I think, more abundantly clear how difficult it is to measure within an actual person this tumor right here, it may be not as easy to tell where it stops and starts. And so a new scoring method to determine tumor size is this tumor volume score. It's a novel method where RECIST takes these linear measurements, and they don't really capture the entire benefit a patient may receive due to that irregular shape I keep talking about. This measurement calculates the tumor volume as a percentage. And so that gives you a percentage of shrinkage. And so in that way, partial response is considered more 50% or more of the percent of the tumor that's there compared to baseline. And progressive disease would be an increase more than 30% compared to baseline. And so you can see from this tumor volume score that if it went from this blue area to the yellow, that's a significant shrinkage. But if we took the diameter, that diameter may be similar. So you can see again how new measurements are coming out that may show and reflect the true benefit of these drugs. And so patient-reported outcomes, or PROs, is another way that we measure whether someone benefits from a treatment or not, or how a patient is just doing overall. So many of you have been asked on a scale of 0 to 10 how much pain you're in, with 0 being no pain and 10 being unimaginable and unspeakable pain. We get used to these type of measurements, and there's equal measurements for TGCT, stiffness, pain, and function. And so for anyone in a trial, they may have filled these out where you get asked kind of repetitive questions about your function, your pain, your stiffness. And then they take an average, and they start to develop a profile of how much people are benefiting in their day-to-day life. So are you able to run errands and shop? Are you able to go for a walk for at least 15 minutes? Are you able to go up and down stairs at a normal pace? Are you able to do chores, for example? And some of these may not pertain to you. And what I found through making the patient registry data is that a lot of times patients are confused, right? That you're like, well, I never did yard work, so how do I answer this question? So finding better ways to capture these very subjective experiences is definitely warranted, but we have to start somewhere. And so some of these PROs is how we started, essentially. And these are validated, which makes them easier to use for researchers, which means that they're not really open to interpretation. They're standardized. And so if we go back to the Tralio study, you can see that patient-reported outcomes, that patients were asked at 9 weeks and at 17 weeks, and then at 6 months, how they were doing. And MRIs were conducted at week 13, as well as at 6 months. And then it was also conducted every 12 weeks after. And so you can see how we're starting to develop a protocol or a schematic for how a trial is run. And so there are current trials in the United States. We have off-label drugs like imatinib and nilotinib, which are globally available. You have Tralio, that study that I keep bringing up because a lot is published on it at this point comparatively, and that is approved in the US. But there are trials all over the world right now. There's AMB05X, which is a joint injection, and that is currently available in the Australia and the Netherlands. Emactuzumab, which is an IV infusion, which is to be determined on where it will be available. You have vimseltinib, which just finished enrollment for its phase 3, and that is available at 32 locations, both in the United States as well as worldwide. And you have this new drug, pimlicotinib, also in phase 3, that's available currently only for enrollment in California at Precision Oncology. And so you can see that this is a very bustling field for such a small disease. And so I'm going to use vimseltinib as an example because, again, the data is published. And so if we look at the study design, you can see that we took TGCT patients. We took approximately 32 patients that were enrolled in the phase 1 study. And there is 45 patients enrolled in the phase 2 study. And so these patients in the phase 2 study were broken up into receiving the drug originally for patients who have never received a drug before. And then you had cohort B had received maybe Tralio or another drug prior. And so they allowed these two cohorts to exist. And so they took a certain amount of patients and they assigned them the drug, and they took a certain amount of patients and they assigned them the placebo, and then they watched them for 6 months. And then they allowed everyone to receive the drug, as well as also their phase 1 study, which is their original safety data, where everyone received the drug and they just captured information on the safety. And so this was based off of originally, and currently the dose is the 30 milligrams twice weekly. And so that's how they started to develop the dose. And you'll see over the course of their life, they started with 30 milligrams every day for 5 days. They started with 30 milligrams every day for 3 days, and then 20 milligrams every day for 3 days. So they started with many options, and that's typical for a phase 1 study that rolls into also a phase 2 study. And so you're just trying to find the dose that you're going to use for that pivotal, really important study. And so currently what they're running is a randomized placebo-controlled double-blind study, which means both patients and providers don't know who received what, and they were randomized to receive it. And where patients received 30 milligrams twice weekly, they were randomized to receive it in a 2-to-1 fashion. So 2 to 1 means out of every 3 patients, 2 received the drug, and they enrolled 120 patients. So that means 80 patients received the drug and 40 received a placebo for 6 months, that 24 weeks. And then after the 24 weeks, everyone received the drug. And so you can see there's trends in how these trials are designed. And so typically now, if I know that a trial is going to be a phase 3 study, I'm pretty sure the design at this point. And so now you guys will also share that same. And what they're looking at, their primary endpoint is the response of the tumor using that RECIST criteria. So how much does it shrink? With secondary things that they look for, which is that tumor volume score we just went through, which is a better representation of the benefit, range of motion, and those patient-reported outcomes like pain and physical function. So for those wanting to know about the AMB05X trial, and this picture's a little fuzzy, sorry, their phase 2 study, their design was a 4-week screening period where patients just found if they were eligible or not. They received the drug on day 1, and they received the drug every 2 weeks thereafter for 12 weeks total. So you have a 12-week treatment period. Then patients are followed for 2 additional weeks to see their effect, and then they have a final follow-up of 12 weeks. So that overall makes a 6-month trial. And so patients were eligible or currently eligible if they're over 18 and they have measurable disease of at least 1 centimeter per that criteria we discussed. In the first trial, they enrolled 11 patients with a single group assignment, meaning everybody got the drug. There's no placebo in this case, and there's no randomization because everyone is receiving the drug. And so you can see that down the line, they're going to have to randomize. They're going to have to probably use a placebo and things like that. And so lastly, I want to go through some of clinicaltrials.gov. Often I get asked, how do I know what trials? How do other people know what trials, and where are the trials? And so I want to go through actually how trials are found and what information you can do. So I want to start here. And so there's multiple ways you can do this. We can just type in our disease. And so I type it in a lot, clearly. So it's one of the first things that come up. And so you can just type in the condition. And so this will give you every clinical trial that's ever been done for tenosynovial giant cell tumor. And thankfully, clinicaltrials.gov is pretty smart, and it knows that patients also search for giant cell tumor of the tendon sheath, as well as pigmented villonodular synovitis, and get these same results. So it doesn't matter which one you look at, you're going to get the same response. And there's 27 studies done. Some of the most cool information I find is just in the status alone, is that you can see what's recruiting. And so you can see that this study, which is done in pexidartinib, so Trialio, is done in Japan. And so they're recruiting to see if the drug works in that population, probably for that regulatory agency. This study is active but not recruiting. And so this is another study for those on Trialio and wondering, can I go off Trialio? This is a study that evaluates going off Trialio and then going back on it.
Sydney, the slides are not advancing.
I'm not on slides anymore. Okay, well, thank you. Let me stop sharing and share my screen. Thank you. Can you guys see me now?
Yes.
Okay, cool. I'm just gonna hit the start over button to make sure everyone's on the same page, because I think this is one of the most important parts. And so conditions, you'll want to type in your condition. As I said, I use it often. So it comes up originally. You have the other searched options. So again, patients search for these together often, so you're going to get the same results. And you have 27 studies done in TGCT. That does not mean there's 27 active studies, though. This is the status option that tells you what status they're currently in. Again, I mentioned that Trialio is being used in Japan, and then it has the sites. And that study that a lot of people are interested in, which is active, which means they're currently conducting it, whether patients can go off Trialio and go back on and see how that affects their growth. And that's being done, as you can see, at 13 different sites, as well as these other sites in the US. Another study that's active but not recruiting currently is the intra-articular or the joint injection for TGCT in the knee, and that's for the AMB05X study. This is an old study done. So you start to see where I get a lot of my information. And very interestingly is they also publish their results on here. So if a trial's been conducted and you want to know, did the trial work? this will have results. This trial was done a long time ago. I think it finished in 2016. And so the results are quite old at that point. But sometimes you can find things that aren't published in the literature. And this is all publicly available knowledge. And so I want to click one so you can see. So I'll click the pimlicotinib one. And so you can see that it's recruiting, that they first posted it in April and they just updated it, and they updated the contacts and locations. So I can quickly see what companies are updating and what they're updating last. And so it gives you the name of the company, and the information we kind of care about is what's the goal of the study. And so the goal of the study is to see if the drug works and if it's safe. And so the drug is given or a placebo for that 24 weeks in part one. And then in part two, everyone is given the drug for another 6 months. And this is considered a phase 3 study. So they're trying to really show that this drug may work in the population they're looking at. And so again, this consists of this part one and part two. It's double blinded, which means patients and providers don't know who received the drug, and they're randomized to receive either the drug or a placebo for part one. And then in part two, everyone gets the drug. And so this is where you get a lot of interesting information. You also get what they're looking for. So in this case, they're looking at RESIST criteria for the response rate, which we know is an imperfect measurement. So what they look at secondarily is tumor volume score, because it maybe better captures what's happening in the actual person. They also look at range of motion, stiffness, pain, physical function, and overall quality of life. So you can see, again, that the designs are very similar across all of the studies. And importantly, the criteria to be eligible in all clinical trials for TGCT currently is that you're over 18, so there's no enrollment of minors, that you have confirmed disease with a biopsy, your disease is measurable, and you're on a stable prescription of analgesics. So if you're taking Tramadol or Tylenol or all of any sort of drug for pain, that you're on a consistent regimen for it, so they know that the treatment itself is helping and you're not just taking more Tylenol, for example. Some exclusions often are whether you're allergic to the drug or its ingredients, whether you've been on prior TGCT drugs, never including, at least so far, imatinib or nilotinib. Also, whether you have significant arthropathy within the joint or severe disease and uncontrolled infection, so things unrelated to TGCT. And then others are kind of more specific to the drug and the person who's applying for the drug, but impaired cardiac function, whether you're using other drugs that would maybe interact, childbearing potential, must use an effective form of birth control. And so you start to see all of these things that would make you consider going into a trial or whether you're eligible. And then importantly, always there's contact information for the sites that are recruiting. Who is the contact for that site? Who is the investigator for that site, and the overall contact for the trial? And so this is kind of the bread and butter of where I get a lot of my information, and there's many pages of it. So you can see also the DCC trial, which is the vimseltinib trial, as well as here, and they all kind of have the same design. This is their phase 1, phase 2 trial, which I presented on a little bit ago. And so you end up getting a lot of information through this specifically, as well as finding it. You can go down and look at, let's start over, and you can see countries. So you can look for your country specifically if there's a site. I find that really important for those that aren't US centric. You can also look at the drug itself. So as you can see, I've looked at every single drug for TGCT at some point. And so if we just look at vimseltinib, we can pull up the trial for vimseltinib itself. And so this is the only active trial with that name. And it is active but not recruiting, as many of us know. And it's the same design where you have that part 1, patients are assigned to receive either vimseltinib or the matching control for 6 months. And then at part 2, patients receive long-term treatment of the disease, but everyone gets it and everyone knows they get it. And so I will start taking some questions now because I don't want to miss any. And so first I see that there's a question that says, how many of the evaluation criteria like RESIST are proposed by researchers versus the FDA? So everything is proposed by the pharmaceutical company. The researchers can advise the pharmaceutical company. There's advisory meetings and advisory members, but the company proposes things. And in some cases, the FDA or regulatory agencies come back and say, well, that doesn't have enough scientific rigor, or we don't think that's going to be the best measurement, or we may not be able to determine that that truly works from this data. And so everything at that point is a recommendation for the approval of the drug. No one's mandated to do a trial a specific way, but a drug may not be approved if you don't follow the recommendations in that regard. And there is a collaboration between regulatory agencies as a whole and those with trials. For example, the EMA, or for Europe, TGA for Australia, all of them work with the company in designated meetings to talk about endpoints, to talk about trial enrollment, who's best for the trial. One hot topic is when kids should be enrolled for trials. There's many rare diseases. 85% of them affects children. So you have to, at some point, treat children in many rare diseases. And so that's a dialogue that happens. So I hope that answers your question. We also had another question about, we are a rare disease, and so funding, how do these trials get funded, right? So there is something called the Orphan Drug Act. It started in 1983. We're just at 40 years from the Orphan Drug Act itself. And that act allowed incentives for pharmaceutical companies willing to work in this rare space. So it gives you a tax credit for those companies, and that tax credit can be above 50% of the trial cost. So that's a really high tax credit. It also waives fees. So like any company, they have to, in order for their stuff to be reviewed, there's something called a prescription drug user fee. And so when they submit something to the FDA to be reviewed, there's a fee associated with reviewing that information. And that currently is around $3 million for a new drug. If you are working in a rare disease, it's waived. You don't pay that. And then what's kind of more lucrative and more appealing is that you have 7 years of market exclusivity, meaning that no one else can come out with a drug for that indication using that active ingredient for 7 years. That's a long time. And so that really is a incentive for those companies. And this was largely because in the 70s, no one was making drugs for any disease in the rare disease space. And there was 200 million Americans at that point, and a lot of rare diseases. There's a thought that 1 in 10 Americans have a rare disease. And so these people are going completely untreated. And so the legislation designated that a disease that affects under 200,000 people in the United States, which was 1% of the population at that time, is considered a rare disease, and you would get these benefits if you did rare disease drug development. And so often TGCT is a bucket, a single little part of a trial in its first infancy stages, and then to get some of those benefits maybe. And then as the drug, if it shows that it works, they pivot and focus on that rare disease. Whereas some trials totally only look at a specific rare disease as well. I think we're just special in that way. Another question was that some of the exclusion criteria for trials are that there's a history of extensive or reconstructive surgery on the affected limb. And so this is largely because all of these trials, their primary endpoint, which means that the primary endpoint is the major endpoint you have to meet to show your drug works. If you fail your primary endpoint, but you show it works on a secondary endpoint, the trial still fails. So that primary endpoint is really important. And the primary endpoint for all of the trials currently are MRI-based measuring of shrinkage. And so if you have hardware because you have a knee replacement or some form of hardware related or unrelated to the disease in the same limb or joint, it's going to make it harder for them to measure the disease. And they may not be able to measure if, one, you have detectable disease, but also if you're benefiting from it. And so that's why that exclusion criteria exists. In reality, I see a lot more flexibility in that. So I wouldn't exclude yourself just automatically. I would talk to a provider before you say, well, I have a knee replacement, I can't go on this. But that's why the exclusion is there. Someone asked if I could talk more about the phase 4 studies. So phase 4 studies are post-market studies. These allow us to understand more about the drug that we didn't capture in the 7 to 10 years it took originally. Drug companies are very motivated to get their drugs out, especially as fast as possible. And as you could imagine, on the flip side, the public wants drugs and they want drugs fast. I know many of us are excited for many of these drugs to come out. And so phase 4 allows information to be captured that isn't necessary for the safety of the drug, but may be more about like children taking it, like how long to go off of it. So that's post-market study about patients with Tralio, and then they're going off and then going back on to see how they respond. That's a phase 4 post-market study to see if patients can go off Tralio. The food effect study for Tralio, whether patients should take it with food and how best to take it with food, that's a phase 4 study. That's post-market. So all studies require some form of post-market research. We call them PMRs per se, or post-market commitments, PMCs. But it depends what information they provide when they go up for approval. And so someone from not the US, if you don't live in the United States, and you're part of, or you're not in, okay, so if you're not in a country where recruitment is currently done, can you participate in the trial? And the option is it depends. It depends the study. Cost is something that's not regulated by the regulatory agencies, at least in the United States. And so some companies will cover the cost of you to travel to that site and all treatment related expenses. In that case, there would be no real reason you couldn't participate. Sometimes there's a language requirement so that you can consent in the language of the consent form. But outside of that, there's no eligibility that would restrict you just solely because you don't live in that country. It may be at your own cost, or it may be covered, and those are very specific to the trial itself. I hope I answered that. And at all times, you can contact me directly, and I can give you more information about that specific trial or any trial. And if I don't have the answer, I surely will find someone who does. Another question is, will there ever not be a placebo required for TGCT? And the question, it's a good question. So I will say one of the things I and in general TGCT support is trying to do with the registry is to make data that is robust enough to stand in the place of a placebo. And so what that means is through everybody answering those questions repeatedly every 6 months, we get a glimpse of how you're doing and how you're progressing through your journey, whether you're on a drug, what treatments you've gotten, and maybe the natural history of the disease. So where many of us seem very different from each other, we may not seem very different if we have enough data. And if you have enough data from enough people that is scientifically rigorous enough, you can say, I know how fast the disease grows on average. I know what to expect on average. So therefore, I don't need that placebo anymore. And so that's the pinnacle of real world data. That's something everybody strives for. And so many of the PROs that you take when you're in a clinical trial, like asking, can you vacuum? And all of those questions we've taken and we've asked them on the survey. And so it's really important, I'll give a shout out, that you fill out the survey and you fill it out regularly so we can promote an environment where placebos won't be necessary. So that's my plug. And that survey can be found in any of the groups as well as repeatedly on the newsletter. And I can post it as well. So if that is all the questions, I also want to thank everybody for their time. We have another webinar on June 15th at 1:30 Eastern time and 7:30, I think it's Central Europe time, CET, in Germany, that area, time zone. And that'll be on the consensus paper as they'll go through all of the parts of the best clinical management practices for TGCT and that overall consensus, which is really exciting. And you'll see that flyer if you have not already. So thank you everyone for spending your evening with me, and feel free to email me at any time.